Okay, so I have 1, 2, 3, 4, 5, 6. I have 8 test tubes for now. How many samples did you each bring? Cedar has 2.
Mine's really old.
Here you go.
You have 1? And you have 1? Pat wants to bring down some Microscope for us to look at, so we'll save 1 test tube for that. So we have 2 extra test tubes if you think of anything else you want to look at. But for now, I'm just leaving these 4 in the middle, and I'm just putting in the center here pipettes, slides, cover slips, and the water that you'll need.
You said that you were going to try and get somebody out to clean.
Yeah, we get a third-party service to clean it because microscopes are usually protected, like all the lenses are protected with a film, like a surfactant stuff. So if you get in there with Q-tips and ethanol, you're just disturbing that protective coating. Well, they can reapply it if it's like a service, but I'm just saying don't yourself take a Q-tip and dip it in peroxide and like rub it all over your...
Dry cotton though. Okay.
You could try, but like if you, like look at this, not ethanol, no peroxide, but like look, you're gonna have to get down in there. See that? So that's why people get Q-tips out and start messing around. The objective lenses, they're the same, like you have to stick it down in there and then outside. So sometimes they'll like blast them with air or, I mean, they have different ways of doing it. So I like being there when they do it, but sometimes I can't, you know what I mean? So I just watch what they're doing. So for all I know, they could be shoving Q-tips in there too, you know, but. Okay, so photo booth on the Macs will get your picture up on the screen. This might be helpful to do in case like we have 3 or 4 people huddled around one scope and that way people can see even when they're not looking through the eyepieces. On this machine, if you'll minimize all that and look for the software called Scopelmage, it should be on the desktop. So do we need any refreshers on how to make a sample? Do you guys feel good about it? 1 milliliter soil, fill it to the 5 milliliter mark, 30 seconds, gentle shaking, 1 drop with the pipette, cover slip, drag it to spread out the droplet, and then drop it on. Starting at the smallest level of magnification. Awesome. All right, don't all jump at once.
Let's do it.
Just remember if you're not using the scope to put the poncho on. Pretty please. Yes, I will take it into my hand. So Cedar brought soil from a greenhouse.
Yeah, this is from the greenhouse, and this is from the mixture of the main fields.
The main fields.
Faded mostly.
Okay. And then you have garden soil?
Yeah, 1 raised bed and 2 just regular garden.
Okay.
Just combined.
What do you have in your coffee cup there?
Pretty much just raised bed material. Well, not necessarily raised, just like an alien bed right now.
Oh, okay.
It's, just,
What do you need? Spoon? Let me get some more.
I believe I should go to 5.
Anybody else need a spoon? You find out real fast who has good fine motor skills and who doesn't.
Do you mind showing how to put this cover slip on? You just set it down and...
So I will take it and, do you mind if I do it?
No, I don't
okay.
mind.
So I just usually like put the edge of it on there and just move it back and forth just to spread it out and then try to drop it. It'll get stuck to your fingers, so you gotta, there you go.
Awesome.
Are you scanning? Yeah, look at that.
It all just looks like little chunks to me, though.
Mm-hmm.
Just take, yeah.
Except, you can't.
Well, it's pretty out of focus for me, but
Yeah. Look and tell me if that's just another chunk or if that's something interesting on the right.
on the right, I think it's probably plant tissue or an aggregate. Mm-hmm.
Yeah, that would make.
At this magnification, you're mostly interested in things that are going to be moving quite rapidly, or they look like monsters.
Wow, this looks so much better than this one.
I know, right? I'm glad that we're a small enough group to where you guys are getting to do this because I was feeling pretty embarrassed about the resolution on the projector. Can you see it?
Yeah, I've not.
Need to place your race.
I'm not sure how to navigate it.
Yeah. Guess what?
Yeah, just gotta play with it, really. Well, maybe focus it up a little bit.
Remember too that you can play around with the eyepieces to get a better focus. On the binocular scopes, both eyepieces can focus. If you turn at the base of the eyepiece, you can focus them. On yours, Kelly, you'll have to focus one at a time because only the left-hand eyepiece has a focus knob. Yeah, so you'll have to focus with your right eye first using the coarse or fine, and then focus, yeah, the left eyepiece.
That was it.
So your different coloration of the particles is minerals, you think?
You're talking about like the brown versus the gold. So those are different organic acids, like fulvic acid, humic acid, ulmic acid. They all have different tints to them. So if you, like when we were looking at the Hogel culture soil, you saw lots of different colors in there. So that's really good because it means you have different types of organic acids in the soil. The darker humic acids are obviously really good because you want humus in your soil, but you also want to have the fulvic acids, ulmic acids, humic acids. There's probably different ones, but they're colored like amber versus mahogany versus black, brown. And I mean, they're just, I'm assuming, metabolized differently by the, by the soil life. So you want diversity there as well as in the organisms.
Like folate, and is it related to it? Mm-hmm. It is?
Yeah. No, that folic. I was saying fulvic.
Oh,
Yeah. Anybody seen any nematodes or?
That would jump out at the highest.
At 10 or at 40x, you would see it like wiggling around. Or if it was taking a nap, you would be like, oh, there's a worm not moving.
What do you know about nematodes? Because we sprayed beneficials for the past 2 years. And they go away in the winter. And why?
Are they dying?
Well, we talked to Pat about it, and he said you have to rebuy them every year.
Yeah.
But I'm wondering, like, are they not native?
I mean, one would think they would lay eggs.
Yeah. And
I
they
don't
actually
know.
go away in the heat of the summer. We use them for flea beetle control.
Oh, okay.
And it works pretty well in the spring and early summer. And then they kind of slowly stop working. That's what we found.
How do they control flea beetles?
The flea beetle life cycle that's in the soil.
So the larva, they eat the larva of the, so they must be, I wonder what kind of nematodes they are. Did you do a full scan?
Well, I want to go.
Like, did you move it around? Oh, you're at 40, you already did your scan. You're at 40x.
Yeah, I don't see.
Well, you're very, it's very crowded.
Are you growing a lot of brassicas, I suppose, since you're trying to worry about flea beetles?
We are.
See if you can find an area of the slide that's less crowded. You may need to dilute that. Wow, that's crazy how much. There you go. Now focus around in there and you'll see. Like I'm interested in that stuff in the background there. Can you focus?
Yeah, I'm wondering.
This is great. From screen to camera, it's pretty good.
It is. We've been working for the past 8 years.
Is this the biodynamic soil?
Yep.
A lot of bacteria.
Well, I'm going to go higher.
You at 400?
Mm-hmm. Looks like maybe an amoeba.
Are you jumping to 100 afterwards?
Looks like maybe a fungal spore.
She's doing 1, but she says she always went to 2, then to 4.
You got rods, you got cocci. A lot of movement in there. Wait, what were you looking at? sorry.
That's okay. And there's three.
A lot of aggregates, a lot of mineral.
I scanned it at the 40X, I didn't see anything jumping out at me.
I'm not really seeing any fungus.
There we go.
The other thing you might see is insect larvae. Little grubs grabbing around. And so that is indicative of anaerobic conditions. Pasture soils, if nobody brought pasture soils.
That was a field that I started on.
Really? Because you could find parasite eggs.
I seen stuff like that.
It was
I
tilled
don't know what that was.
last
Plant material.
spring.
Yeah.
So it was tilled and then worked into beds and then compost applied.
That's what you brought?
Yeah.
Cool. There's a couple fungal fragments in here, but nothing too advanced.
That's a pretty clear picture now.
Okay. You want to look around some more? Kelly?
I was gonna see if she wanted to.
You want to take a look?
Anybody else?
Meredith Leigh, look at this.
Yeah.
There's a lot of what look like kind of clear blobs. You can't see it on the screen there, but I'm wondering if is that mineral? Is that,
The bigger ones?
what are you seeing?
Oh, something just hit it. I see sand
Okay.
particles.
Sand
I
particles,
see,
That's probably what it is. That.
but I also see cysts. You could also have some pollen in here.
Pollen has pretty thick walls too.
Yeah, and they'll have like an oil droplet in it.
Sometimes there's like a red thing. What does that mean if it's like red?
That's a piece of my dress, I think. I don't know.
This is a weird one.
This what?
This one looks like.
Yeah, I mean, you have good aggregation, that's for sure.
I see it over here.
And you have bacteria and you have protozoal cysts, but I don't see any fungi.
Yeah.
Um,
Which isn't really that surprising. I mean, spading would.
break it up a bit, yeah. There's maybe a tiny little piece of a fungus
Eggs together. I don't know.
right there. Hard to say.
Uh-huh. But not one of those spores because it's flat on one end.
Yeah, it looks like it's broken to me. Mm-hmm.
If you move this little diaphragm down here.
Yeah, did you
get some
shadow?
contrast. Okay.
Oh yeah, I haven't
And then
played
you
with
go
that
in and out
one.
like
That's
that, you can
Right
see
here.
them, but they don't look like
You actually
a single
had it
thing.
set pretty well.
I mean, like biological rather. It could be eggs though.
Oh yeah, I see that red bit. What is
yeah.
that?
I'm really curious.
Yeah, there could be eggs.
It's not very clay soil.
It's too big to be clay. It's some kind of silicate particle. I feel like, or quartz or something. It's weird.
Some kind of mineral we have in the soils.
I see that looks like maybe a little fungal fragment right there. I definitely see some. Oh, I think I see a flagellate. Look in there, Cedar. You see the little guy moving?
Yeah.
You can see his flagella kind of coming out. He might be off the field. So the focus is over here. Fine focus is what you'll go for focusing the depth.
Yeah, those are eggs.
What are you guys looking at?
Check it out. It's in the center.
Is this your garden soil? Yeah. Oh, wow. That big thing?
Yeah, it's like a group of eggs.
Oh, the clump.
Yeah.
Yeah, that's some eggs, I would say.
You can see the little, whatever you call it, little egg of what there.
I don't know.
I don't even know either. Why don't you bear
Yeah, it could be bad, it could be good.
That's the tightest, and then just move it backwards and forwards.
So we
It's a
say,
little more
wait,
open if you go to that
does anybody
side.
around here have
You
an
know,
SEM?
not as dense.
Not that I know of. I mean, maybe at one of the universities.
Yeah, I call a couple of them.
There's a fungal spore that looks real good. There's a meat that's a amoebae cyst right there. You've got some Fungal, yeah, there's a real good fungal spore right there.
It's not divided.
Fungal
Do you want to
spore,
come look at it in the,
What is that?
a little bean thing. That looks like, oh, close it out.
but if it's not fungal, what are our other options?
Sand or silt or something?
The rod thing, what is it?
Yeah, like there's.
I think those, they look like fragments of fungal hyphae, but I can't tell for sure.
A lot better.
Just because they have uniform parallel cell walls, and I can see a couple septae here and there, but it's, the smaller they get, the less reliable your visual cues are. But you definitely have protozoal spores in here. That's a good thing. You have diversity of bacteria. Oh, there's a good fungus. Look at that guy.
Wow. Yeah, that's nice.
Ooh, come see, y'all.
Let me see.
Come see this fungus. The other thing is, we said like uniform parallel cell walls, but then if there's a branch, it can have a different diameter from the main stem, and it still qualifies as a fungus. So that's a nice one. Very well done.
Yeah, right.
Now just get one of those in every field, and you'll be fine.
If there's no segmentation, is it plant material, do we think?
You said this is raised.
Yeah, does it look bent or twisted or tattered in any way? It's probably plant tissue.
And there's two places just wrapped the regular garden. One where I intend to plant some okra, and I think when all of them are had some.
It may be a piece of fungus. Like, I can kind of see septae. If you, I'm shadowing differently. Look at it now.
Okay.
Like where the bend is, there might be a cell separation. I'm not sure though.
Moving down there underneath.
Might be good to do in a second. Oh, yeah.
I see some.
Where?
Some of those little spidery looking things.
Below?
Go back.
I see a lot of
Like
them.
right, if you look out.
Right in here? Is that what it was?
That one.
Maybe we want
Yeah.
I see another fungus here that's maybe not so yummy. It might not be fungus. I mean, it's a wide diameter, but it's clear. I would still say it's probably not harmful, but
Well, now, yeah.
yeah, you see it kind of sticking out. It's hard to see.
It is interesting how much sand there is in that one. I don't think that the soil is super sandy.
Well, sand lasts the longest.
I got you some good.
Sand, I'd like to see a piece of what sand looks like.
You wanna come around here?
Please.
There's a lot of sand.
I'd like to see what you mean by piece of sand.
I like this crystalline.
It just looks like chunks of silica, you know, just like clear chunks. Like here and here, refractive.
Almost like a crystal.
Mm-hmm.
Okay. And silt is even tinier than that, right?
Silt is the middle-sized particle.
Oh, that's right, middle and clay.
Clay is tiny. Clay is about the size of bacteria.
So silt,
But silt is quite a bit smaller than sand, you know?
but it still looks crystalline.
It's a little more soft. It can still be, no, it's darker and it's like, I mean, I don't see a lot of it in there, so it's hard for me to describe it, but it's gonna look like softer, rounder, smaller, darker. It's not as refractive as sand is.
I've always thought I had silty soils, but it's like
I mean, maybe you do, because
feel it. It doesn't like feel super sandy.
if you go onto Elaine's site, there are pictures. What's going on over there, guys?
I don't know. We had no clue. We see something.
You see something?
Well, no, just we don't know. I mean, I don't see anything internal.
The amoebas are really hard for me to tell, just because
The
like,
protozoa are the hardest ones.
yeah, they've got
Oh, your sample's drying out. But you have a little more time.
because the sand kind of has a dark membrane. You know, it looks like a membrane just because of the way.
But they're most always going to be pretty round, and the sand is going to be more
Okay.
jagged.
Okay.
So anytime you see like a round thing, you're like, ooh, stop, take a look at it.
So-
What is it?
It
I think it's plant. Yeah. Yeah, definitely. It's cool though. I see some actinomycetes, some actinobacteria right below it there, which is not so great. If you see that here and there, it's not a big deal. If you saw it like that look penicilline fungus there, fungal looking thing is actually bacteria, but it's coming out of that aggregate. And yeah, the question is, look at this, you know, a week from now, look at it 2 weeks from now. Time course samples would tell you if that guy's going to go crazy or if he's just hanging out, you know?
Not look at this slide. You'd have
No,
to
no,
look
no,
at
no,
that
no.
in
It would,
pieces.
yeah, you'd have to go out and sample again.
Okay.
But yeah, there's some more actinobacteria. And little patches of it like that are totally natural. It's totally natural to have it there. But
I
if
have
it's,
a lot I need to go ask my money back.
or you could have done something to the soil to,
Well, it's
you know,
still in my truck, so it hasn't been on anything.
is it in a bag?
Okay. It's covered by tar.
Okay.
So that's not so much a problem for you.
Looks like more actino right there. Might be a little wide diameter, but
Oh, yeah, when I first started, the first thing I did was dump as much organic matter on it as I could.
Yeah.
So the oomycetes are kind of clear and skinny.
and skinny.
Basically, and it's just old hardwood all
Right, I mean,
Tell me if that's an oomycetes.
On a
Oomycetes?
bed.
Oh, my seat. Yeah, I would say, well, it could be. See how it kind of, it's pointy at one end?
Yeah.
It might not be a fungus at all.
Okay.
Some plants have these like structures on their bodies that almost look crystalline.
Okay.
And they can be clear or dark, and they almost look like little hairs sometimes in the soil,
'cause it
and
looks
sometimes
kinda
they'll
like
look like
a
little
hair.
axes.
Okay.
And you might at first be like, oh, is it a fungus? Is it a broken fungus? But it could just be like some kind of plant material that
There's like so
doesn't
much
matter. There's
to
so much.
figure out what.
Yeah, I was gonna see if I could find a pollen.
Oh yeah, because you said you saw that, and I know.
The other thing we could do is,
What's this guy?
it might be a ciliate cyst. I don't know for sure.
Oh, and that is that fungi at the bottom?
Where?
Funny
Yeah,
little
that
structure.
might.
you know, where and just break it into a part
Is
and
there
pull it out.
somewhere
I
we
usually
can
I
find this
got
information
one of the big
online
leaf
so that if we got home, got
You
our
pull behind the
microscopes out, we would know what we're looking for?
And I just get it where the grass. and
Is there a
the
picture of each of the
driveway
things
stuff is.
or?
If you Google pictures of soil microorganisms under the microscope, you can find quite a few pictures. Yeah, often they will.
There's a fantastic poster right over there.
Yeah, check out the poster that we brought you. Buy it from Elaine at soilfoodweb.com or Environment Celebration.
It's up all the microbes.
She's changing it. Environment Celebration now is her site. I'm trying to get a certification to where like I'm teaching her methods to other people, but I'm not there yet. I have to get a good enough grade on my class I'm doing now.
Is it by identifying?
How many classes have you had? You're talking about the online classes, I guess.
Yeah, I'm just taking the online microscope class right now, which is like a precursor to the certification, and it's like 13 courses about morphology and which is a lot of what I've already done. And then there's quantification and there's quizzes. Oh. I think it's plant tissue. See how it looks crumpled? If you look through the scope, you can see in the middle it looks kind of crumpled or smashed.
Yeah,
I just don't see them moving around though.
that makes it.
Fungus will never look tattered or torn like that, you know what I mean? It'll either look broken, like a clean break,
Yeah.
See that right there?
and it'll be really rigid because it's very hard carbon
Yes.
Okay.
structure.
I don't know what that
It's
is.
less vulnerable to breakage.
I mean, the other thing you guys can do is like pull another drop out of your, and have another go because you could pull a nematode out that time, not
Right.
the time before.
Right.
And your sample's drying ananyway, so
Yeah.
I would.
And you were waiting to go, so.
Zoom down in on it.
Oh, I think we're.
Oh yeah, mine's definitely dry.
These are fungal spores.
Nice.
Okay.
And I wonder if that's
So
anything.
how do we clean up?
I've never seen that. It's like a horn.
I can also, if we have some extra time, if everyone feels like you've gotten time on the scopes and you want to look at other stuff, I can show you pictures that I've taken from different things, just to give you more like morphology cues. Really? Is that the biodynamic stuff still? What'd you find?
Something that looks like a horn.
Oh,
And some other long squiggly looking things.
interesting.
Now this is
So that, yeah, that looks fungal. That does not.
right.
Take a look inside there.
Yeah.
It's got a body looking thing.
Oh my. It's a wizard hat.
A withered head.
I'm just kidding. I don't know what that is.
My soil is clay with sand particles. What's that called? Clay loam?
Yeah,
How do you roll it up?
this goes down
Where does that come
here. Now smooth.
I don't know, but when you put it in your hand, it's
Yeah.
got that same clay feel, but then there's big chunks of sand.
Well, you can have a sandy clay. That is in order.
Are we on 400? Yeah.
Is that what
Sandy clay loam.
It's kind of fuzzy instead of like the aggregate. It looks.
This is cool. You got some cool stuff here, man.
That one,
Yeah, that guy like has fuzzy markings.
one of those little spider things.
Oh, the stellate amoeba. Yeah, I want to see one. I've never seen one in real life.
That dark thing in the middle.
It might be, might be a spore. Don't know. There's some fungus, maybe not so beneficial. See how skinny it is in the bottom?
Clear.
Yeah.
Skinny. Well, it's food for something else.
Truth. You can't freak out if you see a bad guy 1 or 2 per slide. It's only if you see them taking over. Yep, You got some eggs in here for sure. Weird. Wonder what they are.
Yeah, I know, probably.
That's actually, I think, plant tissue. See how it's twisted? Right there? You'll have to look through the scope to see it. I'm going blind. I've been looking through the scope for so long. But
That's the bag.
it's a little crunchy right there, but it looks good. Yeah, you gotta mess with the iris diaphragm and the light dial to really get the detail on everything. Does anybody need a clean slide and cover slip? How's it looking over here? Awesome. I think you should.
Soil already nutrients?
Yeah.
Okay, I don't
Well, I
want
have.
I don't want to hog it.
I almost want to like find something super anaerobic and throw it under there. But I'm trying to think of what it would be.
Pasteurizing anything?
Mind your way.
No.
Let me just scoot past you. Sorry. Here goes. Here's a slide for you.
Yeah, they're really. Have you seen those bioreactors that use food waste? They put the food waste in the water and they have these big bubbles on top of it.
Oh.
And the anaerobic bacteria eat the food and it releases methane, which you can pipe off and to use for whatever. And then it leaches down and you get your compost steam. And in the end, you end up with soil.
Bacteria that's,
Bioreactor is
I don't know
called?
if that's what you call it. It's like some kind of, it's for composting.
It's like a
But it's an
biogasser, basically. Yeah, that's cool.
Anaerobic composter.
What's up? Did you find something exciting? I think that's a fungal spore, that little dark orb.
And things like that, so they don't leach out and wash out.
I use a lot of sports.
I think that's why I've heard it's important, is like.
Yeah, I can see cysts, protozoal cysts, flagellate cysts, flagellate cysts, flagellate cysts, probably. That's good.
Sticky and
Are they almost always in the cyst form on this slide? Because they've been through the
More often than not, but like I saw an active flagellate on your sample, and then you didn't get to see it because had my big mug in there, but yeah. But I had to adjust the iris in order to see the flagella, right? At first it was just, but you'll see them, like the bacteria kind of glide around, or they'll sort of, flip and turn, but the flagellates will like bumble sort of, and they'll be bigger than bacteria. So if you see something doing that, then you'll need to mess with your diaphragm to see if you can find a flagella coming off of it. You got a channel of water running through there, so it's hard to see the motility.
Oh, there's that, you see that little thing there?
Did you put one drop on there? Yep. Wow, why is it moving so much?
You wanna take a look?
It's like that same thing that was in mine.
Oh, I don't need, did you look already? Yes, I do want to look. You're at 200. Can I go to 400?
Whenever you want to. I don't know what these little dots mean.
where is, there's another one up there, hyphae.
I think so, yeah.
Has a real thick, fungal hyphae.
Are you making another side?
I just cleaned it, so if you
Okay.
want, I just cleaned that one.
Yeah, you got so much water moving, I can't see the bacteria moving, but you definitely have bacteria. You got rods, you got cocci.
Actually, we're in sea
I see an amoebal cyst. Do you see that in the middle? It's kind of floating.
It moves real fast.
You've been up at Mountain Garden?
Why is everything moving so fast?
Oh, yeah. Yeah, at this point, I'll just come and go.
I want to get to a calm spot, but there is no calm spot.
So you're in the community?
What is that right
I
there?
used to go to you guys,
It's a red.
Sunday,
Flooding in there.
Oh, yeah. It's crazy how much it's moving. Is that like a big old air bubble somewhere?
You guys still doing
See the
the,
segments of
Yeah.
hyphae?
Oh, that little,
Look at this.
that small thing.
This looks like your diagram of that nematode. There's no way. Maybe it just looks like it. Let me see. It's got like a head to it on the right. The brown thing?
Yeah, that thing right there.
Yeah, I can't find it.
Is up.
No, he's gone.
No, it's not a nematode.
Okay. Looks like a round little head.
Serious.
And like striate, like long thing in the bottom.
You ever see nematodes that aren't flopping around?
Yeah, I've seen nematodes that are dead. I've seen them that I thought were dead, but Elaine says they might just be taking a nap.
Had that big swish
Are you involved with the
rainstorm.
permaculture?
I've seen them real,
You know, we don't
going
really
real
know
crazy.
that, it's just in the heat of our growing season, so we're.
What is it? Is it moving around?
Oh, look at there.
Oh, that's a ciliate. Wait. Follow it.
There it is.
Oh my gosh. Maybe it's not. Wait, hold on. Is it just moving with the water or is it moving? That is a ciliate.
Damn, it's gone.
It might be dividing. Do you think it's But I can't believe all that water running through your slide. You might need to pull another one.
Strong current going through there.
It's probably the heat too.
Is that a spore?
That's a fungal spore right there, yeah. I can't see anything.
That's crazy.
Can we do another sample?
Damn it up.
I think it did dry because he waited. He was waiting to use the scope, and I think that's what happened. I think it's just because you had to wait for the scope. So you got dry areas, and then all the water was just going around those dry areas. And would you mind shaking that up for me, and I'll clean this off?
That one had about, I used soybeans as a cover crop
Oh, really?
and
You
then
know,
put
wait a minute, that's center.
hay on it, then the dry or old hay. It had a lot of tillage done, but
Really?
I tried to incorporate as much organic
That looks
material.
interesting. It's fungal.
Ooh, it's got a
So Meredith,
bacterial
I'm
life
curious, we
inside it.
have this greenhouse soil, we haven't tilled in there in like 6 years, but we have tilled.
It's got
I'm
a
really
little
curious
bacteria
if
living inside it.
tilting,
No, really?
how problematic it is.
What's
Yeah.
your,
You can see the bacteria kind of
Like a
bouncing
little
around.
mini-
Yeah, I see some.
It's like a power drill.
I bet.
Or is it just aerated?
It,
Pretty big.
it's like a tiller that goes about this deep and it's run by a drill.
There's a lot of
And so
life right around it.
in the no-till
Yeah,
community, some
rock
people are
is what
like,
it
that's
is.
not tillage, and other people are like, never. And I'm just really curious about how bad or not it is,
Called
because it's
tilther.
What's it looking like to you right now?
Tilther.
Right
the,
now?
Tilther.
What's how's this
Let's
look?
just go in and lift it
Uh,
up.
No, it's just like has your
I don't
horizontal.
know.
Just how much
just
the
move
bacteria is just
in a horizontal
moving
plane, so
around in
like this.
Yeah.
And it
Is there a lot of
just
bacteria?
very top.
Yeah,
Usually
I guess I put on
the
the
bacteria move,
some areas where
right?
they're.
No, they
Yeah,
don't
they will
always.
move.
They wiggle.
Sure will move.
And you see that
I
crystal?
think I need to get closer. 'Cause
What's your organic material in the greenhouse?
Green crystal.
What's your,
What are we putting down?
or what's your percentage of the soil?
Bottom, maybe.
You know? I don't know that offhand.
Yeah.
What a sludge.
It's definitely got our highest cation exchange capacity, but
That's it.
the cation is the most compost there.
So you got really good gas up to
Okay, now I can see the bacteria.
Yeah, make sure you're shadowing. That's one thing.
That really
Yes, I need
helps.
to do that.
We're scanning on.
So one thing you're moving underneath, or two things?
Is it coming through?
This looks very different from there. Yeah, but you're still
That
up
one's got one
on
of those.
But he also, like I said, stood around with that slide for a while. And I think all these aggregates sucked into these certain parts of the slide, and then all the water was channeling in between them. And that's why we couldn't see anything. So scanning, I don't see any big guys. So you guys want to do this? You're welcome to.
That one. That's a big, fungal.
All right.
Big.
I mean, ever since I've been using a microscope, I can't even imagine doing it.
Totally.
Oh, there's another one.
You need some help?
Down, right there, that guy.
I just want to find another ciliate. Find one of those ciliates. And we'll show everybody.
Yeah, there's a lot of bacteria. Also mineral hygiene.
You can also dilute more cedar if you want to pull it up to 10, 1 in 10.
Okay, what does that do? Just help you see.
Just spread it out a little bit more, help you see it.
Yeah,
actually see that.
Yes.
The other way, other way.
A little bit. It doesn't look like you're necessarily a good one. It's kind of clear.
Well, but it is wide.
Okay.
The oomycetes will not be that wide.
Okay.
So it's probably not a bad guy. Probably not the best guy ever, but he's not harming anything. He's putting good structure in your soil. And
Right.
Hold on.
also,
I don't know if that's.
Also, I mean, ever since I saw those bacteria sucking all the cytoplasm out of fungi, I've often wondered if I see a wide diameter fungus without any color to it, is it just a dead beneficial fungus?
That's what I say, the other way, because we can't say right or left, 'cause
Yeah.
it doesn't, it's,
Well, the fungi will kill off their own hyphae too. They will
Yeah, if it's not
doing it- if it's not doing work for
them.
attach it. Yeah, but it's basically a carbon storage source.
Yeah, it's
Which
doing
can be
you a
consumed
favor
later,
in,
yeah.
in terms of structure.
Uh-huh.
Totally.
I heard you over talking about mulching with leaves.
Mhm.
And that bed was initially mulch. It's a garlic bed. It has
Bacterials
pieces
here.
in
I
it.
don't.
Yeah, that's
Of
what
course, these are the best thing for
I do
drugs.
see, like, if you look in there
Well, I
right now,
stuck hay, I
it's
put
like
about 3
a
inches of
little
hay
bit
on
left
it.
of center,
Yeah.
I see some
You can mulch
flagellate
it with,
cysts.
and then when the hay is
Good.
here, the worms
It's that quick.
yeah, and then I was like, it's
Here.
February, and I was like,
It's hard for you to see because it's not.
where's the dirt coming from? So I got like 20-some cubic yards of
Well, that leads me
leaves
to
are
another
here.
thought, which is
edge of the
we
field.
do a lot
And
of
so I went down there and started
carping.
put it down there that thick.
Yeah, I put them on 6 to 8 inches before.
I wonder if you're
And I
going
don't miss
anaerobic
that.
a little bit with your
And it's like-
tarpon.
Yeah, it's a lot of rods in there.
But I don't see like actino, and I don't see lactobacillus, and I don't see,
I'm real pleased at how this.
like, that's a
the leaves have really
cyst.
suppressed the leaves-
See that?
the weeds because in February.
Right there is definitely a cyst, yeah.
What's the cyst again?
There's a guy right there.
Beginning stage of the sprout.
What's the cyst again?
They were-
That's definitely a cyst of some kind, probably amoebae or maybe,
Another one.
Sunshine comes up the dirt.
Yeah.
So I
which one?
covered up to try to drown them out.
This one?
Like that.
Well, you see how it's jagged?
I'm really impressed with the leaves.
I've been growing garlic and leaves for,
It usually has 2
40
walls,
years,
you
I
can
guess.
tell, like,
and
Yeah,
I've had, I grow a lot of elephant garlic.
Okay.
But I've had.
There's also like things inside of it, look. if you,
yeah, I was
like-
looking at this one right here.
That little one?
Right there too.
Yeah, I think that could be pollen, or it could be, see how it has that little like, it's either an, but it could be an entrance or an opening too, like it doesn't, yeah, it just depends on which direction it's facing.
Presence isn't very useful, it's really not much of something you see.
No, there's some-
I guess it could be.
Looks like a little algae there.
You know, look at the dots.
Well, they look pigmented to me, which is why I said, isn't that so fun?
Those bacteria are just so
There's fungal spores right there.
So maybe organic materials are some fungal.
Those are green lemons, actually.
I keep thinking I'm gonna see a flagella
That's straw in there last summer, so we
on one of
bought
these.
tarps and straw around the tomato.
What kind of vegetables are you growing?
Every kind.
Yeah, everything. There's tomatoes and peppers last year, and then in the winter, we crop it constantly, no peppers. What are these guys?
Well, I was trying to focus to see if it was a fungal
Spinach, I mean,
hypha or not, but I can't tell.
there are a lot of brassicas and chenopodiums in there. And don't they prefer?
Brassicas don't use, actually, one of the few plants that we have.
Flip this thing in.
Yeah, and I heard that chenopodiums.
I just didn't know if I need to go down.
Yeah, they
Also,
don't
that
either.
chenopodiums don't either, so there was a lot of spinach and beets and chard.
I got it.
Oh, you got it?
Yeah, I just didn't know if it's another stage went down.
Arugula, so,
A lot of annual plants.
Is this your soil? Yeah.
but they don't like kill off
Still like,
mycorrhizas.
no, they don't, and they like extra mycorrhizal stuff too.
Oh, okay, that's interesting.
So, there's ecto, endos. They're not actually mycorrhizal until they develop a relationship with the roots. There is, you know what I'm saying? Like, otherwise, it's just a fungus.
It's just hypey.
Yeah. Until it's actually.
You have such good aggregation though, Cedar, that it doesn't seem like it could possibly go that anaerobic. You know what I mean? Like
Yeah.
maybe pockets, but
Breath work in there. And we don't step on, permanent. It's 8, 7 years old.
I'd love to see that at a 1 in 10 dilution.
Okay.
Because
do
my feeling is that you have protozoa in there. It's just hard to see them because there's so much going on.
So just add more water to
Go
this,
up to
bring
the
it up to
10, and then pull
Make a new
another
slide.
slide. How's it going over here, guys? Do you find any ciliates for me?
You can look, I don't know.
It's actually a good thing if you didn't, so.
Oh, well, good.
That was just really exciting. There's some fungus.
No, I see it there to the right. Well,
There's a bunch of those like
yeah.
that. There's at the edge of the slide, and you go over to the edge, there's a bunch of, there's one that has several branches in it.
Where?
We got
At the edge of the
That is
slide,
annoying.
down to the bottom.
I thought we had one there.
Bottom left.
The one that was on it was busted.
All the way almost off the edge of it.
Over there, I'll get one.
Sciencing.
Is there something in there?
I thought that was it.
I think there's another part of this box with the cover slips.
You got a lot of fungal spores in here. You got a lot of protozoal cysts. You got a lot of bacteria. It's good. It's good looking stuff. That's a weird red. Weird. Yeah, that's good segmentation. It's kind of narrow, but it's got an interesting color.
Wood chips, aroma wood chips.
There's clear. Oh, I thought they got clear thing.
Yeah, that's not a fungus. That is like plant tissue or something.
Well, I saw this is really good too.
It takes such a long time for it to break down, don't it?
Not really, not the sawdust. I mean, if it's mixed well.
Okay. Well,
There you go. There's, see, the thing is, you're gonna
Well,
get different.
I'm sad we didn't find more ciliates, but it's good news for your garden.
Yeah, right.
Denser woods. So
This one's stiller.
really kind of jack up your. It does
Well, that one was huge.
That's the lid. Here's the.
Oh. Right in front of us, we're like, oh.
It's kind of hard to see things the same way when you're looking at microscopic things. You're like, yeah, y'all go crazy with that, or pull another one, or whatever you want to do. And I can also pull up some pictures, but I'll need that computer. The other thing I've done is like, if anybody makes sauerkraut, it's really fun to take like the brine of your kraut and look at it under the scope. So you'll see anaerobic conditions in there, right, under the surface. A lot of lactobacillus. Or like beer or wine that's fermenting, if you want to look at yeasts, which are anaerobic fungi. So
Are they about the size of the bacteria, or are they different?
they're more the size of the protozoa, and they're more oval-shaped, yeasts are. So you wouldn't want to see a bunch of them in your soil, but in your beer that's brewing, great, you know. But if you end up getting a scope and you're like, how am I going to familiarize myself with all the morphology? Like, find things that you know will have yeast in them or lactobacillus and look at them, and you'll learn that way, you know what I mean? Yeah. Looking at anaerobic compost teas or swampy soils or soil that has been like left in a closed container for, you know, airtight container, and then look at it, like see what you see. And you learn more from finding bad things than from looking at good things over and over and over again. You know what I'm saying?
Yeah.
need a
Differentiate.
sheet with I guess you can print something like that off later on, or we can, I mean, but like the different identification and.
We can't see things move.
Yeah, there's not a place where it's like, this is the best, this poster that we have is the best sort of aggregate of all the different organisms, but you'll have to sort of mess around online to find. It's helpful to find online not only microscope pictures, but also drawings, like illustrations of the different taxonomic groups, because then you can kind of get a better idea what you're supposed to be seeing, like where the flagella are occurring and what kinds of internal organs. I just do Google searches like microscope pictures of protozoa and see what comes up, you know? And a lot of times Google will randomize it and you'll see like streptococcus and you're like, well, I know that that's cocci. Coccus is a bacteria. I don't want that. But you can find, you know, pictures. And if you haven't had a chance to look at this, come look at it because...
Study slides.
Oh, a couple years ago. This looks different than the one.
The bacteria look bigger in this
More like a
case.
pussy.
Mm-hmm. Yeah, but is it because she believed it so much that?
So this is the kind of stuff we won't see today, like rotifers and stalked ciliates. Those are more like anaerobic, microarthropods. Those are cool. How's it looking?
There's something interesting over here. That's got
great.
fuzzy look to it. And things are sort of chowing down on the edges.
Oh, really? You think they're eating it?
Yeah, totally. Yeah, they totally. Oh, you mean on that thing? Yeah, it looks like it.
It's the way it's kind of fuzzy on the edge, that black.
It looks like things are sort of going at it.
Oh, you're right, I hadn't noticed that.
Weird, what is that?
I saw one of those on the other side. One of those fuzzy edges.
I mean, it's not a spore. There's no cell wall. It could be pollen, but yeah, I would say that'd be my vote, is that it's a pollen of some kind. And that the reason things are hovering around it is because it's got bacteria stuck in it and they're, bumbling in and around it, or flagellates are going over there to eat, but
Yeah, it looks like it's got bacteria inside it, unless it's just over or under, I'm not sure. Can you, maybe if you shadow it a little bit or something too?
the unfortunate thing about the map
There's another one. It's
Yeah.
just, it's no longer on the computer
Right
screen.
there.
You can see a bunch of, look like little bacteria.
Yeah.
Yeah, right there.
Yeah,
You see it?
there's one there.
If you look
and
up,
there.
it's different here. It's not, it's hard to tell because you can see the margins more on the
Yeah.
scope than the screen.
Kinda looks like this guy over here.
When you see a lot of flagellates, that's another indication right there.
ciliates are more of an indication of anaerobic. Flagellates need good oxygen to do their work. If ciliates will insist if there's too much oxygen for them, and it's actually more likely at that point that they'll die than to exist, right? So, exist. A lot of cocci, a lot of bacteria in here. You got some fungus. You got some. I'm sure you have some protozoal cysts in here. It's just, yeah.
I'm just thinking about weeding it though, like if the dirt kind of gets in the pathway.
There's a weirdo, bad guy.
Weirdo?
Yeah,
No.
that little white thing.
It looks a little tattery to me to be a fungus.
Oh, okay.
Plant.
I mean, it might be. It might be oomycete. Oh, everything's moving.
Yeah.
Around inside out.
Where? In the middle?
Yeah.
That looks like a piece of sand that you're looking through it
Is
to.
that what it,
Go all the way up to the corner real quick.
Yeah, the way to know for sure would be to tap that cover slip with a toothpick or a needle or a pen or something gently. Where are those stellate amoeba? I want that. It's going to be a riveting video of everyone staring into the microscope.
Serious editing.
That is a cell wall on
The black thing in the middle? Or it's in the middle for us?
To me,
There's a lot going on here.
be clear like that? Is that what sand looks like?
Sand, yeah, it's like, and it'll be refractive. Like you'll see the light sort of bouncing off of it in different angles, and there'll be jagged edges.
So that's gonna be
Yeah,
sand.
that's kind of sand particle or some kind of silicate crystal or something like that.
No, that right there.
Yeah. That looks like some plant tissue of some kind. Same with that right there.
Okay. So we're looking for like really round with a cell with a wall around it.
Looks like a fungi, I don't know.
Oh, that little flat.
Yeah, it's really long.
It's probably not the good
It
kind.
looks like fungal.
Yeah, it's dark.
It's not
The
sick.
bacteria are just
Yeah,
small.
it is.
Or rods.
Hang on, you can see it really well in here.
See the rods too?
Yeah, okay.
I don't know which one you're talking about.
And it's really long.
You can see it better. Yeah, you can see it better in the scope. But yeah, they're just elongated. You'll see more ovular looking bacteria too that are kind of, they're not quite rods, but they're more oblong cocci. They're called pediococci. It's different genera too, so that's good. Looks like you got some cysts in here. That's good.
What type of soil do you have? Sandy silt, clay?
I think it was so long.
So silty sandy?
Yeah.
I'm going cross-eyed looking at all the different scopes that are adjusted for people's different eyes.
My neighbor shoves all his to the edge of the street, and burns it every year.
Maybe it's got a little
And they come by and
like
they
sand
directly
particles
across the street. It would take a lot of wheelbarrow loads.
So this is segmented.
Oh, cool.
See this?
Yeah.
That looks
There's
like a
a-
nucleus in there to me.
That looks like some fungus right there, for sure. For sure.
And that looks like a piece of hyphae right beside it.
You think that's hyphae? That thing that just has.
like a big old
turned up.
Which one, this black?
It's kind of got that like crunched in the middle. It might be plant material.
But see that one in the middle?
Yeah. What is?
That's
It
pretty
looks like
good, you
a
guys.
nucleus to me in the middle of it.
Mm-hmm.
Isn't it too fuzzy on the margins though?
Where's this in?
That's probably a fungal cyst too, or it could be an
If
amoeba
it's dead, they
cyst.
actually kind of
Double wall done.
Yeah, it
they get
does.
glomerulin on them. There's another one. What's that? That's either a spore.
Cool. I guess that would in there and fall over.
Then there's stuff inside of these aggregates too.
Yeah.
I think you definitely got cysts.
I think it's one of them.
Cross-eyed over there.
Ah, that's it.
Maybe we can find something in the refrigerator to look at.
Which, what really helps is like to get stuff that has.
This one of those.
You get used to seeing it.
Oh, yeah, like the maker.
No, one of those bad.
You could probably, take
You
a
know what to look for. Like get it
Do you have any mushroom honey?
anaerobic. I don't do much.
No, it is a bad.
I mean,
Meredith, what's the bad
we go for walks, so I observe what's around, but I've never
You said it's like
gotten
a
really into it.
Oh, my.
What we do sometimes.
Is that man? It's just like a clear. No, I'll leave it alone.
It'd be really
We look at
skinny.
pictures of the ones that we want to look
Yeah,
for.
yeah,
And
yeah.
then we just
So
sit
you
and
can
meditate
sharpen
and
your
sort of
eye.
like, it
Yeah.
works. It works.
See that?
And then you go find them?
Yeah, it's a lot easier to find that.
I don't think that's the worst they get right there, but it's definitely narrow, but it's darker in color, which is a good, and I see pretty regular septi, so
Narrow things out.
Yeah, because there's a lot of things.
I don't think it's terrible.
Maybe.
At least the rate.
And it's not in a mass, like it's not growing in a mass web like that picture we looked at. That would be concerning. Yeah, that'd be like, oh man, we need to aerate. Yeah. But it's, no, the one we just looked at, I wouldn't classify it as a bad fungus. I wouldn't say it's the best, but like we said earlier, it could be there just providing good soil structure and, retaining carbon, and that's a good thing, but it's not necessarily do anything else,
How small can this cyst be? Like how, could that potentially be?
Yeah,
Or is that too
definitely.
small? 'Cause that's almost just a
It
little
looks
bit
like a
bigger than
flagellate
bacteria.
cyst to me.
Okay.
Right here,
Yeah.
that's way bigger than bacteria.
Wasn't that bacteria next to it?
Yeah, but
bouncing
most of
around?
what you're seeing there is a shadow.
Okay.
Like if you were to
Yeah,
focus
it's
on
like-
that.
It's the surface tension around it. It's like twice as big as the bacteria.
It's addictive, isn't it?
It's a very small world.
All right, I'm going to take this computer from you if that's okay. And we'll hook it up to the projector